Hi everyone, in this video we will explain the process of synaptic transmission and how the neuronal cells communicate with each other. So the title of this video, Neuronal Chemical Synapses. Synapse is the site of conjunction between two neuronal cells, by which the neuronal cell would be able to send its current to another neuronal cell via chemical substances called neurotransmitters. The neuronal cell that is responsible for releasing neurotransmitter is called presynaptic neuronal cell. The space between two neuronal cells is called synaptic cleft, and this space is about 20 to 40 nanometer in width. The neuronal cell that receives the neurotransmitter via receptors on its surface is called the post-synaptic neuronal cell. Now let's look at the process of synaptic transmission. As we mentioned earlier, a neurotransmitter would be released from the presynaptic neuronal cell to the post-synaptic neuronal cell. This process is so sophisticated in that one neuronal cell could branch off and form 2000 synapses. But here in this video we are focusing on the general concept of synaptic transmission. Now there are three types of synapses. A synapse could be a junction between an axon from the presynaptic neuron to a dendritus in the postsynaptic neuron, and this is called an axodendritic synapse. It also could be a junction between the axon of the first neuron and the cell body of the second neuron, called an axosomatic. Or it could be a junction between an axon from the presynaptic neuron and an axon of the postsynaptic neuron, and this is called axonic. Now let's look at the process of synthesis of neurotransmitters. The synthesis of neurotransmitters starts in the nucleus, and then some modifications are done through the endoplasmic reticulum. Then the synthesized neurotransmitter will be packed in vesicles when it's passing by the Golgi apparatus. These vesicles would be transported via microtubules through the whole length of the axon. Then it will be stored in the synaptic knob, ready to be released whenever a signal is coming by. Now let's look at the process of neurotransmitter release from the synaptic knob. Here's our synaptic knob. The first step is the arrival of action potential. When the area of the synaptic knob is depolarized, there will be an opening of voltage-gated calcium channels, causing calcium ions to enter the cell down its concentration gradient. Calcium ions will bind to the vesicles and stimulates its movement. And then vesicles will move and fuse with the cell membrane, releasing its contents into the synaptic cleft via the process of exocytosis. Now the excess amount of calcium will be pumped outside of the neuronal cell against its concentration gradient via calcium ATPase pump found in the synaptic knob. You can also notice that mitochondria are found on the synaptic knob for providing energy for the process of neurotransmitter release from the presynaptic neuronal cell. Now that the neurotransmitter are released from the presynaptic neuronal cell, so what would be the fate of these chemicals? There are four ways in which neurotransmitters could go. First off, they could bind to receptors found on the surface of the postsynaptic neuronal cell. Some of these neurotransmitters could be degraded by enzymes found in the synaptic cleft into their basic structural components. Or they could get absorbed back to the presynaptic neuron via the process of endocytosis, or they could diffuse to the capillaries found around the synapses in the extracellular fluid. And now let's look at some electrical events taking place on the post-synaptic neuron. First off, a neurotransmitter binds to receptors found on the surface of the post-synaptic neuron. These receptors are connected to a ligand-gated channels. Electrical events happening could be either excitatory, in which partial threshold depolarization on the cell membrane is taking place. It could be due to sodium or calcium ligand-gated channels, transporting positive charges to the inside of the cell down their concentration gradient. This process is called excitatory post-synaptic potential, and it's a type of local potential. Electrical events taking place also could be inhibitory, in which potassium ligand-gated channels transporting potassium ions to the outside, or chloride ligand-gated channels transporting chloride ion to the inside of the cell, causing a state of hyperpolarization, making the cell less excitable to stimulation. Now let's look at how the action potential is initiated in the post-synaptic neuron. Action potential is initiated via the summation of many local potentials, and there are two types of summation. First off, temporal summation, in which a repetitive stimulation to the post-synaptic neuron occurs over a period of time. Or it could be spatial summation, and here there are many presynaptic neurons giving signals to one post-synaptic neuron, so that excitatory post-synaptic potential happens on many sides on the surface of the cell membrane. All of these excitatory post-synaptic potential will be summated, and an action potential will be initiated in the area that we call the axon hillock. And as we mentioned in our previous video, it's the most excitable part because it has a lower threshold and a huge number of voltage-gated sodium channels. And then action potential will be propagated through the whole length of the axon. Then let's take a look at the process of inhibitory post-synaptic potential. First off, a neurotransmitter binds to a receptor, causing a state of hyperpolarization, and this is called a direct inhibition. Indirect inhibition also could take place, and it has two reasons. For example, the post-synaptic neuron is on the refractory period, in which this cell would be refractory to stimulation, so no action potential could be initiated. Or it could be due to third neuron acting on preventing the presynaptic neuron to release its neurotransmitters, and this is called presynaptic inhibition. Now just a side note, GABA-aminobutyric acid is a well-known inhibitory neurotransmitter found in almost all parts of the central nervous system. Now that we are done with the electrical events, let's look at some properties all synapses share. First off, the synaptic transmission is a unidirectional process from presynaptic neuron to the postsynaptic neuron. Second, the synaptic delay. It is found that when signals come to the synaptic node, there is a delay of about 0.5 milliseconds until the neurotransmitter reaches the receptor of the postsynaptic neuron. And this is helpful for counting how many synapses we have on a specific part of the central nervous system by counting the sum of the synaptic delay. The synaptic fatigue. After repetitive stimulation, the vesicular stores in the presynaptic neuron are depleted, or the receptors on the postsynaptic neuron are inactive, So no synaptic transmission will occur at this time, and this is called the synaptic fatigue. Then post-tetanic facilitation, and this takes place after the synaptic fatigue happens, in which there are many calcium ions on the presynaptic neuron, so there will be neurotransmitter release without signal arriving to the synaptic node for a brief amount of time. And the concept of convergence and divergence. Convergence is when many presynaptic neurons converge into one post-synaptic neuron. Some of them might be excitatory and some might be inhibitory, and it's for controlling the signal's output. On the other hand, divergence is when the axon of the presynaptic neuron branch off and spread signals into many post-synaptic neurons, and it's for spreading of information throughout the nervous system. And now let's look at some factors affecting the process of synaptic transmission. First and most important factor is the presence of oxygen. Synaptic transmission is an active process using aerobic glycolysis for producing energy. The brain could last for only 5 seconds before entering a state of coma, and this is how important oxygen is for the signal transmission throughout the nervous system. Second factor is the body pH. A high body pH, which is called alkalosis, increases the activity of synapses, and that's why with alkalosis, patients tend to develop convulsions. A low body pH or acidosis causes a significant suppression in the synaptic activity, and patients with acidosis tend to develop a state of coma. And now let's look at some drugs that might affect the neuronal synapses. There are so many drugs working on the synapses. Here we are just taking some examples. So for example, let's take caffeine and let's see how it works. So in the central nervous system, there are adenosine receptors, and this for a neurotransmitter called adenosine. When adenosine binds to its receptors, it will promote the feeling of being tired or being sleepy. And what caffeine does, that it binds to these receptors and block them. So when these receptors are blocked by caffeine, they could not be bound to adenosine. So there will be no feeling of being tired or sleepy. Instead, there will be a feeling of higher activity. Let's look at another example, which is the local anesthetics. For example, let's take lidocaine as an example. There are pain receptors in the human body which are free nerve endings. These, when a pain-producing substances or neurotransmitters for pain come for these receptors, they will conduct these signals and send them to the brain. Local anesthetics works by blocking sodium voltage gated channels, and by blocking sodium voltage gated channels there will be no propagation of impulses, and when the signals are not propagated through the neurons, it will not reach the brain, so there will be no feeling of pain, and the pain signals would not reach the brain. Now, before we end up with this video, there is a special type of synapses. It's called the neuromuscular junction, and it's a synapse between the neuronal cell and a muscle cell. And that part of the muscle cell which is connected to the neuron is called the motor end plate. In the muscle cell, we can find these invaginations which are called the subneural clefts, and these for increasing the surface area. These invaginations contains the receptors for the neurotransmitters released by the neuron. So an electrical impulses come, an action potential comes, and then the calcium binds to the vesicles and releasing the neurotransmitter from the neuron. The neurotransmitter released to the muscle cell is acetylcholine, and the receptors on the muscle cell are nicotinic receptors. These nicotinic receptors are bound to a ligand-gated sodium channels. When it's activated, they will promote the entrance of sodium to the muscle cell. Sodium entering into the muscle cell will cause a state of subthreshold depolarization. It's called the end-plate potential, another type of local potential. When these end-plate potentials are summated, they will initiate an action potential, resulting in muscle contraction. In the muscle cell, unlike the neuronal cells, and the end plate potential is always capable of producing action potential because of the huge amount of acetylcholine released from the neuron. Now, regarding factors affecting the neuromuscular junction or drugs acting on the neuromuscular junction will be discussed separately on the chapter of musculoskeletal system. So that's it, guys, for today's video. Thank you for watching, and until the next video.